All right, hi everybody. It's Dr. Chrissy. Thanks for coming to GeneBites today. I have my team with me, my team from NGI. I have Michelle Godfrey. I have Laura Kelly. Say hi, and I have Samantha Menz. So today we're going to be talking about sulfur intolerance, and my CNS, Michelle, has already recorded a GeneBite for us on sulfur intolerance, and it's actually in our presentation today. But so we're not going to go through and watch that, but we do have a quick little PowerPoint put together for you guys today. But the GeneBite Roundtable is what we're calling this. We're basically going to hang out with you guys and kind of go through this presentation, but you're going to get to see us in action and just kind of hanging out and talking about these pathways. But like I said, we're really going to be focused on sulfur intolerance and transsulfuration, and specifically the CBS Upregulation SNP. So I'm going to go ahead and share my screen, and we're going to kind of get through this pathway. And hopefully, if you guys have any questions, you can throw them in the comment box, and we'll be looking forward to answering your questions. So we'll go ahead and get started with this PowerPoint. All right, see if we can get the PowerPoint to load. Technology is always fun. There we go. All right, so here's our presentation from Michelle. And like I said, hopefully it won't try and send us to YouTube. There we go. All right, so I'm actually going to have Michelle kind of go ahead and talk to us a little bit about the patient's perspective on sulfur intolerance. So one of the things I wanted to ask you, Michelle, is, what exactly is sulfur intolerance? There's a lot of confusion about that. So like, I mean, if you go to Yellowstone and you breathe in the sulfur fumes there, does that cause sulfur intolerance? Are we looking at sulfur drugs? I mean, what exactly is sulfur intolerance?
Well, I know firsthand that it's not breathing in sulfur gases, because I was just in Taiwan and I went to a sulfur spring and I have a sulfur intolerance. So I got out of the car and started breathing, and I thought I was going to have a reaction, but I did not, luckily. So sulfur intolerance is when you can't process sulfur well and it builds up in the body and you start having some of these reactions. And this can be from food you're eating, mostly things like cruciferous vegetables, or if you're eating a high protein diet. One of the biggest things I see in people is having garlic breath. So if your husband has really strong garlic breath after eating, that can be a sign that he may be eating too much protein and not processing his sulfur well. Brain fog, sometimes you have anxiety. That's a whole pathway of how that's created. When you have memory issues, or you have sulfur smelling gas, that's a big one. So those are some of the things I look for when I'm looking at people's symptoms.
Okay, I definitely think there's a guy at the gym that has sulfur intolerance, because he basically sweats out garlic next to me every day.
Yeah, or that ammonia smell, you can tell, the guy at the gym also, yeah.
Yeah, it's it's always, you don't want to say, hey buddy, you want to come in and give a swab and we'll take care of that problem for you, or maybe he's taking the garlic pills or something like that. So, one of the things that I think a lot of people come to NGI for is maybe, they think that they have, they have autism spectrum disorder, that kind of thing. And so, a lot of youngsters, will have or be on the spectrum. And we do often find that they do have sulfur intolerance. And so one of the things that I found pretty remarkable here is, when we do have active CBS upregulation, that they almost always do have like the GI disturbances, which is kind of crazy. They end up having like this, and this is kind of gross, we're talking about, stinky gas and that kind of stuff, but their stools, they actually look like wet leaves. They have like this really weird consistency to them. So, for for patients listening, trying to kind of figure out, we're talking about these microbiome imbalances and GI disturbances. They can be pretty remarkable. Sometimes they even call them like rice water stools. And the other thing that we typically see sometimes is, in older folks, we do really have, that extreme sort of brain fog. And some people even get diagnosed with Alzheimer's or dementia. So pretty remarkable on either end of the spectrum when we're talking about, high levels of ammonia in the body. And we're going to go through the pathway and kind of look at these things, but it's pretty remarkable little gene or enzyme as we're gonna learn here. So, all right, so we will kind of go on to the next section here and learn a little bit more about how this is actually functioning in the body. So, as I mentioned, we're talking about enzymes. So genes are proteins and proteins are enzymes. So to kind of simplify this a little bit, most enzymes are downregulation. So this particular sulfur enzyme is an upregulation, which makes it pretty unique. It's a little bit different. So, but to understand the downregulation, we're talking about basic chemistry. We've got a substrate or a starting point, a cofactor, which you can kind of think as a catalyst. It's not a true catalyst for our science buffs out there, our chemistry buffs, but for people who aren't terribly familiar with it, just think of it as a catalyst. It basically makes it go. And then we have a product. So our substrate, our cofactor, and our product in the downregulation, we're essentially going to have a low product because it's basically going to stop this process here. And then we're going to have a high substrate, okay? And the cofactor may be variable here. This is, likely going to be low because we don't have enough of the thing to make it go. But with an upregulation, we still have a substrate or a starting substance. We still have a cofactor, of course, in our product, but our cofactor basically is going to get, used up because it's going to go really, really fast. Then we're going to have a high product and a low substrate. So when we start thinking about how we validate this enzyme, trying to make sure that we're looking to make sure that it's turned on. So we have a bunch of symptoms that we look at, but we're also making sure that, for our clinicians or our doctors out there who are looking to see whether or not this works or not, this is a pattern that we're going to be looking at very specifically. For this one, it's homocysteine over here and cystathionine over here. So some of our statistics here, we're really focused again on this upregulated variant. We've listed it here if you are out there biohacking and looking at your own genetics. But this is very controversial. So we don't have a lot of literature to actually support this gene. And that doesn't necessarily mean that it isn't so. It just means that there isn't a lot of published research out there. So clinically, we see this a whole lot. Because as you can see on that first slide that we showed you guys, there are lots and lots of things that are out there that are associated with this. So I'm going to throw this over to my team members here. Laura, how many patients have you seen or clients have you seen that have had CBS upregulation or that you can think of off the top of your head?
Right, a lot.
A lot.
A lot of people seem to have this. Yeah. And part of what I see too, and I know we were talking about a little bit, is that you can have a couple of the different variants too. And so it's trying to figure out which one's actually expressing. And so actually a good example is my dad, who has both, and he had high ammonia. And so it was obvious that was the upregulated variant was actually the one that was, more active. And the other thing too is I notice when I do give vitamin B6, we have to be really careful about how much we give because you can actually push the sulfur pathway too. And then you're like, oh, oops, okay, let's take that down a little bit. Kind of went too far too quick. But it's a good way for us to see how our supplementation is actually working. Well, so that's another way too, if you don't necessarily always have the genetics, that you can kind of be looking at it.
Yeah, that's exactly right. So, I mean, we're not really focused on CBS downregulation today because that's not part of the GeneBite that we're looking at or the snippet that we're looking at today. But if you were to have both variants, because, I mean, if you think about SNPs, if you think about a football field and again, I'm not really a sports person here, but this is an analogy that most people get. If you think about a football field, that's the gene, and then the yard lines along the field, those are actually the SNPs. So, I mean, you can have multiple SNPs within a gene. And so, it is possible that you have both a downregulated variant and an upregulated variant. And unfortunately, if you have one of each, that doesn't necessarily mean that they cancel each other out. So, this is something that is really important to keep in mind. So, as Laura mentioned, you can push a pathway too much by giving too much of a cofactor. And the other thing to think about is vitamin B6. It works on a lot of different pathways in the body. So, one of the things that we'll talk about here in just a minute is that vitamin B6 is also the conversion factor for glutamic acid decarboxylase, or GAD. And it's going to drive the production of glutamate to GABA. And so, while this could be a really good thing in some cases, it's also going to be driving this conversion factor here from homocysteine to cystathionine, which is going to produce ammonia. So we have to be really, really careful as we are trying to balance things in the body. In terms of our cofactors. One of the things that, I kind of get on a soapbox at SNPED is that nothing acts in isolation. So, that's getting nods from everybody around here. So that's a big thing to kind of keep in mind. The big take home for CBS is that it's important for the production of glutathione, taurine, and sulfate. So as again, we're thinking about our validation aspects, those are the key markers that we're going to be looking for. All right, so pathways, okay? So some people are visual learners, other people like to see words. So essentially transsulfuration is a fancy word for homocysteine catabolism. And if that still sounds like Greek, it's basically a pathway that's breaking down the amino acid derivative for the final methyl acceptor for MTHFR. And I kind of laugh, I'm kind of being silly here because that also sounds like Greek to most people. So, homocysteine is essentially, we're adding a methyl donor into this system here. Methionine is up here. Over here, if we have the other picture, this is, a methyl donor creation system. In one carbon metabolism, methylation is essentially a methyl donor recycling system. And homocysteine catabolism is essentially a methyl donor elimination system. I call this the garbage disposal system. And so we're essentially trying to get rid of the methyl donor that was created over here in one carbon metabolism with MTHFR. And we're trying to ultimately get us to glutathione over here. This is our primary target. We do have some
really, I actually think this is really important too, because a lot of times when we're looking at MTHFR and people think of that, they're sort of thinking of it in isolation of this piece and thinking that this is how we influence like homocysteine levels, is through MTHFR and not just through transsulfuration as well. And so it's like putting that piece together a little bit there.
Yeah, it's super important to think again that literally nothing acts in isolation. So at Omics, which is our gene panel company, our little software company, we work on algorithms that go up and down on either side of the pathway. So, you really can't, and we look at things that are, 5 to 10, sometimes 15 different steps above and below one particular genetic variation. So you really can't just say, oh, I have this one SNP, I'm going to treat it right here, and I'm expecting that it's going to change. That's really not the way you practice nutritional genomics. It's really, really important to make sure that you're not only validating the one marker that you're looking at, but also up and downstream of that particular location. So super, super important. All right, so again, transsulfuration, just to kind of make sure that we're all on the same page here. This is the only true disposal route for homocysteine. Okay, so homocysteine gets a bad rap. It's bad, it's inflammatory, it's going to cause heart disease. This isn't really true. It's kind of an intermediate place, okay? It just so happens to be something that's stable in the serum and we can measure it. So homocysteine has a lot of different influencers. So kind of try and reset your brain and not necessarily think of homocysteine as a bad guy. If it's elevated, it does mean that there's pathway impairment, but it just doesn't mean that it's, that it all by itself is a bad guy. The other routes that are in this pathway, in the methylation pathway, so methionine synthase and betaine homocysteine methyltransferase, that's what these abbreviations for, they basically just recycle homocysteine. So this one has a cofactor of B12, this one has a cofactor of zinc. So, we really need to be working on those two cofactors along with vitamin B6 if we're trying to get rid of homocysteine. So again, the whole purpose of transfer sulfuration, we're trying to get glutathione, taurine, and sulfate. And then glutathione, of course, is really important for detoxification. So this is our master internal antioxidant in the body. Okay, this one looks a little bit more complicated, okay? And this is why we're really talking about CBS upregulation today, or that sulfur intolerance, okay? So CBS upregulation, so coming over here and looking at this, homocysteine, again, is going to be low, right, if we have upregulation, okay? So this is going to go zoom really fast through here, okay? And we're going to use up all of our vitamin B6 here, okay? So if we were going to look at this, we're going to have low levels of B6 if we're going to measure this. Okay, so CBS upregulation causes what we call sulfur dumping, okay? Now that sounds kind of gross, but again, remember how we talked about the GI issues? It's one of the reasons we call it that. But it also biochemically dumps this, okay? So we have increased cystathionine, we have increased taurine, and increased sulfate, which is the sulfur dumping aspect, okay? So without kind of going through the complicated biochemistry here of all of this, just understand that as we push through this pathway, we're also pushing down through here. Unfortunately, what ends up happening is we end up skipping this important part over here that creates glutathione, which is a bad thing, okay? We need glutathione. Again, this is what protects us from cancer. It protects us from detoxification issues. And then unfortunately, what ends up happening is that it starts to dump ammonia into the system. Ammonia is neurotoxic, and this depletes something called biopterin. Do any of my team members want to tell me about biopterin?
Well, BH4 helps make our neurotransmitters. So that's why sometimes when you have this, you're going to have anxiety or depression because you're not making serotonin and dopamine correctly.
Exactly. What stands in for biopterin in the one carbon metabolism cycle?
Folic acid.
Right.
Right. So when people take methylfolate, when they have CBS upregulation and they think they feel better for a little bit, what are they doing, right? They're replacing biopterin, but then after a little while, they also feel more
anxious,
right? Because the methyl donor is actually contributing into this pathway. So a lot of people go, I've got CBS upregulation, I have MTHFR, I'm going to take, methylated folate, and it helps for a little bit because it helps to replace their dopamine and their serotonin artificially. It's like a giant Band-Aid. But it doesn't actually address their ammonia issues. So they still have brain fog.
I just had this situation happen too with somebody I was talking with, and they said that their mom was getting heart palpitations in the morning and like a burst of anxiety. And, she was on methotrexate, and so they were giving her methylfolate. And so we kind of went through that, and it was like, well, you should maybe try a different form of folate because the methylfolate was very likely causing almost like a panic attack. And so this person switched that up, totally changed everything. And his 93-year-old mother stopped having panic attacks in the morning. So wow. It was a big like,
Yeah, so a lot of people are using the methylated vitamins incorrectly in so many cases. Yeah. And if they do have MTHFR, they're likely inducing folate trapping on top of that. So really and truly, they should be taking something like adenosine cobalamin to address, up the chain to correct for this imbalance here. And so unfortunately, if they do have CBS upregulation, they're not addressing their ammonia issue, they're not addressing their glutathione deficiency. So essentially it's increasing their risk for things like cancer or other, detoxification issues, which is pretty amazing. So they get a slight benefit, and most people can kind of, I call it standing on your head. If you get a 20-30% increase in the way that you feel, and you go, man, I don't feel as bad as I did, and they go, okay, well, I can handle that. And then they stop trying to optimize on the way that they feel. So it's enough of a Band-Aid to keep them from pursuing and actually correcting the underlying biochemical pathways, which obviously has pretty severe consequences. All right, so what do we actually find with upregulation of CBS? So we're going to find that we have decreased vitamin B6, and so for our clinicians or for our biohackers, you're going to validate this with a marker called xanthionate. This is something you would find on an organic acids test. Typically, you're going to find homocysteine that's going to be low. This could be a blood test or an amino acids test, and of course, glutathione you would expect to be low. Another organic acid that you could look for would be called pyroglutamate. You could also find amino acids, glycine, cysteine, and glutamic acid might be low, as those are the building blocks of glutathione. If you are actually assaying biopterin or BH4, there are some tests that look at the ratios of these. Then you could find, or BH2 to BH4, then you could see that this would also be low. Patterns, and this is all about pattern recognition, guys. Increased, you would find elevated sulfate, ammonia, nitric oxide, cortisol, glutamate, and taurine. Now, glutamate, again, is something that's really interesting, something that we find a lot in foods. So what are some of the foods that we find glutamate in? Sam?
Anything with the notorious food additive monosodium glutamate or MSG.
Okay. Yeah, so MSG is something that, unfortunately we still find a lot of. Sometimes it's really sneaky. A lot of times they hide it in foods by calling it autolyzed yeast or hydrolyzed yeast. So, we find this in a lot of our, packaged foods or anything. A lot of our vegan foods have this kind of autolyzed anything in them. And as I mentioned before, so our enzyme glutamic acid decarboxylase, it uses the same coenzyme, vitamin B6. So if we're deficient in vitamin B6 because we have CBS upregulation, we're not going to be converting glutamate into GABA. GABA is a calming neurotransmitter. It's the same neurotransmitter that we kind of tap on when we take things like Xanax. So we're going to have incredible amounts of anxiety when we are looking at elevated levels of glutamate. In fact, we call this the panic attack gene.
One of my favorite things, by the way, is to have a conversation with people about this when it's like, well, how do you know which foods have glutamate in them or a derivative or something like that? And I always say it's the food that does the party in your mouth.
Yeah,
right? Like that mouth taste, because it's doing the same thing to your brain. It's basically overexciting the brain too. And it feels really good, essentially at first, and then you just get too wired, or it's the kid that goes to the birthday party and eats all these foods and comes home and is, yeah. It's not always just the sugar. A lot of times it's the additives that are causing that problem.
Absolutely. The other thing here is elevated ammonia. So, for my medical doctors that may be listening in, most of the time you think of ammonia, you think about having to do like a line in to actually measure, ammonia levels. In the functional world, we look at organic acids testing, and we look at things like orotate and citrate and isocitrate. So those are more metabolomics markers. And so when we find all three of those to be elevated, we must always see that this is an elevation of ammonia in the body. The marker citrate has another really interesting association, but not necessarily when it's elevated, but when it is lowered. And we're actually going to be talking about this next week on our next GeneBite. Anybody want to pop on and tell me what low levels of citrate means?
High oxalates.
High oxalates, that's right. So hyperoxaluria is typically associated with kidney stones, but it can also be associated with things like fibromyalgia, also with dementia or Alzheimer's, interstitial cystitis, all kinds of things. So we will have a nice deep dive into our next GeneBite in terms of oxalates next week. So you guys make sure you join us for that. That one is really, really prevalent. A lot of people have issues with oxalates. Not as many people have issues with sulfur sensitivity. Typically, if you have issues with CBS upregulation, you're going to know it. All right, so moving on here. So there's one other thing that we can, that is sort of what I call guilty by association with CBS upregulation, and that's sulfite oxidase or SUOX. This SNP is basically below in a CBS, and this is a sulfate gene or sulfite gene. One of the reasons that we look at this or even talk about this is that there is a coenzyme called molybdenum, and we actually use this to kind of take some of the pressure off. So for people that actually have active CBS upregulation, we give them a little bit of molybdenum to help with the processing of this. So true SUOX SNPs are very, very rare. Homozygous SNPs are very, very rare, and they cause neurological abnormalities in infancy. So you're most likely not actually going to see these in any of your direct-to-consumer testing or any of that kind of stuff. But you may end up seeing, mild presentation or heterozygous SNPs here. But when we're looking at this, we're basically looking at the conversion of sulfite to sulfate. And again, if you have someone who has active CBS upregulation, you really do want to give them a little bit of molybdenum to kind of take some of the pressure off of that pathway to kind of shift the conversion back up to glutathione. So again, our lab validation, we want to see typically decreased homocysteine below 5. We're often going to see increased xanthuronate, increased sulfate, and increased taurine. The last thing that I do want to kind of mention here is that we didn't get a chance to talk really about homocysteine, but if you have joined SNP-Ed or have looked at any of our advanced lectures on folate metabolism, folate and what I call extracurricular folate metabolism, which is essentially all of the other methylation capacity, not just the methyl donor recycling center of methionine to homocysteine. There are lots of influences on homocysteine. So homocysteine is not a really valid marker. If you have really, really active CBS, it will influence homocysteine no matter what. But you may have mild variations of CBS upregulation, and you may have somewhat normal homocysteine. We're really trying to get between 5 and 7, as Michelle mentions in the GeneBite. So I do encourage you guys to listen to that as well today. All right, and yeah, making my point there. All right, so the intervention. So I'm actually going to have Michelle go ahead and talk about this, since she talks about this in our GeneBite here. So Michelle.
Sure. This is just for a short period of time because sulfur is important. This is not a long-term diet. This can be anywhere from 2 days to 2 weeks until we get this under control. So we're going to reduce your cruciferous vegetable intake. This is garlic, onions, broccoli, cauliflower, that kind of stuff. Protein, all protein, eggs, meat has high sulfur content, so we're just going to cut that down some. And then we're going to add in the molybdenum, which I can never pronounce, molybdenum, to help support that SUOX enzyme, which will help clear the sulfur from the body, and then we'll slowly start reintroducing these foods. Oh, and another important thing is sometimes you're getting B6 in places you don't know. So we might look at some of the other foods you're eating that are high in B6, or if you're supplementing with a multivitamin or other formulas. That's why we don't usually do multivitamins or formulas, because you may be getting way more than you think you're getting, and that may cause that enzyme to work a lot faster than it should. So that's where we would start, and then we would assess your symptoms, and then maybe redo your labs.
Yeah, exactly. Exactly. Anybody else have anything else they'd like to add? No? All right. Well, yeah, again, it's dangerous to stay on a low sulfur diet for a long time. So this SNP and multiple other SNPs are found on our optimal diet gene panel. You can learn more about this at the Nutritional Genomics Institute webpage. And thank you guys for joining us today at GeneBites, and we will see you guys next Friday at noon. And again, we'll be talking about oxalate sensitivity then. So we will see you guys then. Keep learning, and we'll see you soon. Take care, guys. Bye.